Yeah, now we are on.
Thank you, Leandro. So, good afternoon, everyone. It is a great pleasure to introduce you Javier Moreno, who is a colleague from the CONICET in Argentina. Javi, I will make a short introduction about your career. So, Javier got his undergrad and PhD degrees at the University of Buenos Aires. During his PhD, he described the molecular basis of the plant growth defense trade-off in the lab of Carlos Majari. I don't know if I'm speaking correctly.
That's fine.
Then he moved to Michigan State University to work with JAS repressors of the jasmonate pathway upon supervision of Greg Howe. In 2013, he went back to Argentina to work on plant transcription factors controlling plant responses to environmental clues. So during the last 5 years, he was using two model plants, Arabidopsis thaliana and the liverwort Marchantia polymorpha, to study the evolution of AD-ZIP transcription factors and their relationship with biotic and abiotic responses or stresses. This research group recently discovered the biological functions of oil bodies in liverworts. So, Javi, thank you very much for accepting our invitation. As I explained, I probably you have like between 45 minutes to 1 hour to make the presentation. Then we will open questions from the students, from us, and also from people watching via YouTube. So thank you very much, and you can start.
Well, thank you, Igor, Magda, and Leandro for this kind invitation. Let me share my presentation first. Tell me if you can watch it. Can you there?
Yes.
Okay. Well, thank you. I'm really happy to be here. You know, I was also helping ASPB in a seminar series that we were doing during the pandemic, and I was really hard to bring people from South America there, no? Because sometimes it's hard to get those spots. So I really find this opportunity part of that, you know, try to strengthen our nets, local or regional nets, to strengthen our research programs. So I will present you here our most recent work on these really fantastic chemical factors that are called all bodies in liverworts. So I will tell you a little bit more about it. But first, let me show you an outline of my talk. I will talk a little bit, you know, about the plant transition from the seawater to land. I will talk about the traits and especially about the associated genes required for that transition. And I will specifically talk about the case of the HD-ZIP transcription factors. That is a family of TF that I'm studying. To finally, you know, develop my talk on our results and our conclusions. So as you can see here, this is a general graph showing you in the x-axis time. No? These are million years before present. So if you move from your left to the right, you can see mainly how complex is getting the organization of a plant. You know? And basically you move from unicellular organisms to a multicellular plant to finally, you know, once it's done the colonization of plant maybe 400 to 500 million years ago, after the colonization, you have the big radiation of plants that take place maybe 200 million years ago. So you can sort of easily guess some of the traits that plants require to move from the seawater to the land, okay? So, and that's maybe the main question here, okay? What were the traits and what were the associated genes to those traits required for this transition here, okay? So, and the general feeling maybe 10 to 20 years ago was that this transition to land must have been accompanied by gene innovation, okay? And that was actually a reasonable hypothesis at that time. And at that time, I'm talking about 10 years ago, we didn't have the data to answer it, okay? But that was our best guess, let's say, before the high throughput genomic era, okay? So then it started to came out the results of two big projects that actually are, you know, expanding the way we try to approach these questions. One was the 1KP transcriptome project that ran from 2008 to 2018. Like the name tells, it's, you know, they wanted to sequence the transcriptome of 1,000 species, okay? But the singularity of this approach was that they didn't want to go further into crops. They wanted to get transcriptomes from different plant species, talking about from unicellular plant species growing in the seawater to multicellular or even pluricellular species, okay, growing on land. But the point was that they had to belong to different plant lineages. And so the white paper of that project came out in 2019. That was the cover of the Nature journal. And that data is, you know, heavily being used by multiple labs in the world because it's public. Now there is another big project running that is called 10KP that actually is not, you know, it's actually trying to sequence not only the transcriptome but also the genomes of 10,000 species, again spanning a wide evolutionary time. These projects were received mainly the support of the Canadian government through the University of Alberta and the Chinese government through the Chinese Academy of Sciences and BGI, that is, you know, you have probably heard about it because it's the largest sequencing company in the world that has, you know, even their own, they are developing their own sequencing technologies. So for the first time in the history of biology, we are gathering an important volume of data coming from these programs, giving us the chance to answer basic questions related to the transition of plants from the seed to the land. And again, this is possible thanks to new sequencing technologies and novel bioinformatic approaches to manipulate this huge amount of data. So I will give you just one example of what kind of things can we do with this. It's a beautiful paper from Liam Dolan's lab. Liam's lab has worked mainly trying to identify genes, well, let's say Arabidopsis genes controlling root development, okay? But in this work, they ask how many families of transcription factors we can find in different plant species spanning a wide evolutionary time, okay? And for that, they use the 1KP database. So I want you to pay attention here, no, to these numbers circle with within red circles. You can see that here it moved from 17 were present. So this is the number of TF families found in rhodophytes, 28 the number found in chlorophytes lineage, 39 in the charophyte lineage, 47 in liverworts, well, sorry, bryophytes, 47 in lycophytes, and finally 48 in angiosperm plants. Okay, So, but if you focus specifically within the number of families that they found in land plants, you see that actually the number didn't change a lot, okay? So maybe, so now we can say that there was not much innovation, okay, in terms of gene families. Maybe the innovation came later, you know, with gene duplication events. And I will talk to you, and I will show you about that data, about that later, okay? So we can conclude, this study concludes two things. First, that there is an early origin for most of the TF families, okay? In that sense, okay, most of the families took place before, has an early origin, okay? And second, that if you look at these numbers, 17, 28, 39, the stepwise increase in TF family number is actually before radiation of land plants, okay? So again, the main conclusion is actually that there was not much innovation of new families of transcription factors in land plants, genomes, yeah? So I will talk to you today about this family of transcription factors called the homeodomain zipper, and that's they are called like that because they have a homeodomain fused to a leucine zipper domain. This gene family was actually heavily studied in angiosperm plants, in solanum, in sunflower, a lot in Arabidopsis. And what so far the main role of them is their function in the abiotic stress tolerance in those plants, okay? And some of them actually were used, well, one of them coming from sunflower is actually licensed to in soybean and wheat to generate drought tolerance in those crops, okay? So I was working on that family of transcription factors when I came back to Argentina. So there was a question given rounds, you know, that we were wondering about the function of if this function was also conserved in non-vascular plants, okay? And so if this function was part of an ancestral trait, you know, conserving the most common reason ancestor. So what are liverworts? I have to tell you that, no? What and why we are interested in liverworts. So liverworts, as I mentioned you before, are non-vascular plants that together with mosses and hornworts form a lineage called bryophytes. So they are not economically relevant, but ecologically. First, because they form, you know, they are part of a tropical net. No, they suffer herbivory. They feed other guys. They establishes interaction with fungi, with other plants. And second, because they are, well, they grow in the understory of forest or in different habitats. And third, because they are usually first colonizers after big environment disturbance, okay? So in that sense, they can be very useful to restore environments that were heavily disturbed. So as I mentioned you before, bryophytes split from flowering plants more than 400 million years ago, okay? That is important because that divergence can give us an insight into the past and how plant diversity has evolved, okay? The first fossils, I didn't mention that, the first fossils of land plants actually belongs to a liverwort and has almost 450 million years of age, okay? So this is a field trip that I did recently in this area. These are little plants actually, And the plants that you see is actually the gametophyte. So that's a big difference with angiosperms, okay? Because the generation that you see and is the more prominent in the life cycle of the plant is actually the haploid generation, okay? I will talk about you later about these gemma caps or these structures that are produced there. So in those structures are actually produced these clonal propagules, okay? So basically, it's a mechanism of asexual reproduction where a plant can produce clones of itself, okay? The interesting thing, maybe the most outstanding characteristic of this lineage, of liverworts in particular, is that it's the second largest group of land plants after angiosperms with more than 8,000 species. And from the biochemical standpoint of view, they are great because they have a huge chemical diversity. No? So actually, there are up to now like 1,600 compounds that are only found in liverworts. So you can imagine the potential in terms of pharmacological or biotechnological use that these species have, and they have been barely studied, you know. And so some interesting thing about these liverworts is that several compounds, chemical compounds of these guys that are actually interesting are actually synthesized and accumulated within these oil body cells that contain an oil body, and they are called like that because they have oily compounds. But their function was still unknown, okay? So within the liverworts, there are a huge variety of oil body cells, okay? So in oil bodies, you have different shapes, color, texture, number per cell. So in the case of Marchantia polymorpha, has one large cell. It's a specialized cell that develops only one large oil body, okay? But you have, for example, here, a species that develops these little organelles that are oil bodies and has several oil bodies in every cell, okay? What is interesting is that this oil body diversity is also associated to the oil body biochemical profile of that species, yeah? And so you can identify, again, multiple compounds that are specific of different group of plants, of different groups of liverworts. And some of them are actually further studies for example, in the case of Marchantinae, that is a cyclic bis-vivencil. It's being used in tumor control. And for example, Radula has, so far is the only liverwort that has cannabinoids, and has a cannabinoid that is actually 1,000 times stronger than the one extracted from Cannabis sativa. No? And then, as you can see here, you have fatty acids, you have terpenoids, and so on. So we propose here to use Marchantia polymorpha, a liverwort plant, okay, non-vascular, remember that. Remember that the plant that you see is actually the gametophyte. The advantages of Marchantia polymorpha is that we have the genome fully sequenced since 2017 with almost 19 protein coding genes. And the good thing for us, molecular biology guy, is that it's easy to transform using agrobacterium. And one of the most interesting things is that it has a reduced signal transduction pathways, okay, particularly ETF. So that means that the number of TF, not the number of families, the number of genes within each family is really low. No? And I will show you this table with some of the families, not all of them, but some, comparing Arabidopsis thaliana with Marchantia polymorpha. So, for example, you have almost 100, more than 100 genes of the Matchbox family in the Arabidopsis genome. However, in Marchantia, you have only 2 copies, okay? One of each belonging to different subclasses. The same happens for the HD SIP 1, 3, and 4 classes, 1, 2, 3, 4, that only has 1 copy of each gene, while in Arabidopsis has almost 60 in total, okay? And that's actually at the genetic standpoint, it's very useful, you know, because you can not only get knockout mutants in the whole class, okay? Because again, if you knock out this mutant, you have a plant that doesn't have any MIC-C gene, no? But the other thing is that you have, given that it's the gametophyte that you are transforming, you have right away the plants, the transgenic lines that you can use, okay? You don't have to select for, you don't have segregation effects, given that you don't have the homozygotes like we had in Arabidopsis. The other thing that I want to mention here that is really frequently in Arabidopsis, tomato, and other species, angiosperm species that we use, is that, you know, you don't have the problem of genetic redundancy. No, that means that is several genes doing the same at the same time. You know? Sometimes, in order to see a phenotype in Arabidopsis plant, we need to cross multiple times to get a higher order mutant in order to see a phenotype. Here, it's really can be really easy in terms of technical issues that you may have. Said that, we wanted to answer this question. No, is the C1 HDC ancestral function also related to abiotic stress tolerance? Okay. So we proposed using Marchantia polymorpha to answer this question. Okay? So with this, I'd like to move forward to my results section. So I will show you mainly our last 2 papers. Okay, so one was actually using the YKP transcriptomic data. Okay, using the 1KP, again, you have a wide array of plants, of species belonging to different lineages. And you can see here, you know, in gray, light and dark gray, liverwort and mosses. Okay, those are bryophytes. And what we found here and we describe here is that actually all land plants have only one inherited only one copy of the most recent common ancestor. Okay, so that means a couple of things. First, that all the copies, all the gene copies that you observe in other species, land plant species like angiosperms, are coming likely from the application events. No, and that is important because the application events sometimes and frequently actually leads to new functions, okay? And the other thing is that given that Marchantia has only one gene copy, we can actually test if that only copy has or not the same function like the one observed in angiosperm species, okay? So with that in mind, we were able, you know, to generate mutants of the C1HDC gene using CRISPR. So we took different advantages, different strategies, sorry. In 2 cases, we we changed the open reading frame of the conserved domain and the other two are whole deletion of the gene or deletion of the whole conserved domain. No? We had 4 lines that we were using along our work. And so far, you know, it was quite, I have to say, that we try multiple abiotic stresses like drought, salinity, mannitol, cold, freezing, UV, high radiation, starvation, and in all those responses or those stimuli, the mutant plant behaves similar to the wild type. Okay, and well, that's something, no? When you have a very nice hypothesis and you get the opposite result. So, like here, for example, this is plant weight, this is fresh weight of the plant, and you can see here plants growing in controlled conditions, they do have the same fresh weight, but after salinity or mannitol treatment, they basically respond the same way. We also, given that most of these responses are also centralized by hormone in non-vascular plant as well as in vascular plant, that is abscisic acid, we made an ADA dose response to test the germination of gemma, and basically we didn't find any difference between the mutant and the wild type. So again, this was a little disappointed, but when you don't have a phenotype, you try to find another. But this was actually, you know, mainly this was rejecting our main hypothesis that we had at the time. So we moved we went back to the mutant and we started to look at it and try to find something else. No? So as I mentioned you before, the plant of Marchantia polymorpha developed these gemma caps, and within them there are asexual propagules called gemma. Those gemma, and also in the tale, but in the gemma are really apparent and easy to observe, are these highly refractant cells that are the cells that contain the old body. You can see them, you know, either in the male or female gemmas, okay? And here, this is Nile red. Nile red is a staining that binds to hydrophobic compounds like lipophilic contents, and you can see the oil bodies are heavily stained by Nile red. When we looked into the mutant, basically the mutant doesn't have those whole cells. We didn't observe them, neither when we stain with Nile red, okay? So this is a new phenotype. The problem is that we didn't know what were the oil bodies doing, no? So to confirm that our transcription factor is responsible for the formation of the oil bodies, so again, this is the wild type, where here we are using body pad to stain the oil body cells, and you can see these strong green dots that says that this is the mutant, and this is actually the mutant complemented with a wild type copy of the gene fused to a fluorescent protein, the TD tomato, driven by the native promoter. Okay, and the cool thing about this is that effectively, you know, this the expression of the wild type copy fused with the TD tomato was able to restore the development of the oil body cells, and well, the body pie clearly indicates that they are also filled with hydrophobic compounds. Okay, it's just the quantification of that. So here we conclude that this TF is actually required for oil body formation. Okay, so then this time we were collaborating with a group of Milton scientists at the Max Planck Institute in Cologne, and because they are heavily working in HDCP controlling the leaf formation in Arabidopsis and cardamine, two angiosperm species, but they were also interested to answer this question, you know, if the Aegis SIP was able to control similar traits. And they found an interesting correlation. So first, they observed that the gene is heavily expressed around the apical notch, so around the meristematic area, okay? But actually, if you quantify it, if you quantify the expression is even higher when you get a little further away from the notch, okay? And this is in pink, you can see here the expression level of the gene. And at some point, at some distance, you start observing the first all bodies differentiating, okay? And that can be seen here with these green dots. That, if you observe carefully, those green dots actually co-localize in some cells with cells that are heavily expressing the transcription factors, okay? So that was a good correlation. So how can we say that actually that you need the expression of the transcription factor to develop the oil body? So we thought about this experiment. You know, this is a cell tracking experiment. It's called cell tracking because at the T0, so the initial time, you identify a cell that is highly expressing that is peaking, let's say, you know, highly expressing the transcription factor, and then you follow that in the time. Of course, you need for that a good confocal microscope and a good facility to do that. And then, so for example, you follow the same cell, okay? And so here, for example, the same cell after 12 hours developed the oil body, okay? Only the cell that was highly expressing the transcription factors, okay? So with that, we were able to say that the C1HD SIP expression peak precedes oil body formation, okay? So we did some more quantification, no? Because if you look at here, you can see that around the notch, you have different expression levels. Around the notch is high, it's higher in the middle region, and the further you move, the expression only remains high in those cells that develop an oil body, okay? So we conclude a couple of things. One, that you need expression peak, and that expression peak precedes oil body formation. And for some mechanism that we still don't fully understand, the expression fades away in neighboring cells. We don't know, we still don't know how. So, of course, what do you do? Well, when you have this kind of, you have a nice tool here that is a mutant that doesn't have any, that lack oil body cells. So we compared the transcriptomic profile of the mutant against the wild type one, okay? And we knew for a previous paper from a Japanese group that non-axenic conditions, so growing the plants on soil or even starvation, induce the number of oil body cells, okay? So we basically grow plants in axenic and non-axenic conditions and compare the mutant against the wild type, okay? So basically what we were trying to see here is those genes that keep downregulated no matter what condition, what treatment or growth condition we're growing on, okay? And within this selected group, we found actually a couple of transcription factors like MIB2 and NAC6. We found CYP12, and I mentioned this here because later on was described that the mutant of CYP12B develops, lacks also the development of oil bodies, oil body cells. And more importantly, maybe in the context of this biochemistry class that you are taking, within this group, there were several terpene syntheses, okay? Like TPSL2, it's a gene that you can find in angiosperms, but liverworts also have these terpene syntheses that have a microbial origin, okay? And several of them were downregulated, okay? So there were reports, previous reports, that within the oil body cells, some of the compounds that were accumulated there were terpenoids compounds, okay? So we wanted to go and confirm that, confirm that the murine was actually lacking, not only show reduced expression of terpene synthesis, but that also synthesize and accumulate less terpenoid compounds compared to the wild type. Okay, and that's what we actually did. So we basically prepare extracts of these plants growing in different conditions. In this case, we grew the plants in starvation, and we compare them with the wild type. Okay, so let's observe first this the panel from the left. Here, so these peaks here are all of them sesquiterpenes, and you can see here in black and gray are wild type tissues, okay? And you observe that, well, you have several peaks that belong to sesquiterpenes. However, in the mutant, there are the blue and light blue peaks. Those peaks are actually missing or very low, okay? Then I will show you more detail on them. However, and this is nice, that it's not that all the peaks are gone, this is specific for sesquiterpene compounds. So for example, the D-terpene phytol is present in the wild type, but also in the mutant, and the same happens for several fatty acid compounds, like these peaks 8, 9, 12, and 13, okay? So this was done by GCMS and GCFID. And in this case, as I mentioned you before, these plants, these extracts were prepared with hexane. So, for example, here is the similar experiment by comparing specific compounds grown in controlled conditions, non-axenic, so plants coming from soil and starvation. Yeah. So, for example, for 2-hydroxine, a sesquiterpene compound, you see that in control plants, the levels are very low. When you grow the plants in non-axenic condition on soil, the levels of the 2-hydroxine are induced and are even higher induced by starvation. However, in the mutant, are the blue dots here, the blue bars are basically depleted in 2-hydroxine, the same for chamigrine and other sesquiterpene compounds. It's the only one of the few exceptions about the monoterpene was limonene, that is actually abundant in liverworts, that shows a similar pattern for that, that is induced by non-axenic condition and by starvation, but is absent in the mutant lines. The opposite case is for phytol, as I mentioned you before. Basically, phytol is not induced by these treatments, and the levels are pretty similar to the control condition. Okay, so with this, we have enough data to say that our transcription factor, the C180CYP, controls the expression of terpenoid-related genes and also the terpenoid content in the plant, and is likely associated to the lack of oil bodies. Okay, so but so far, what is the biological function of C180CYP if it's not related to abiotic stress tolerance? No. So here we came back to the angiosperm information that we had. There is a lot of publications relating terpenoid contents to herbivory or stress in general. So we decided to test the biological function, through the side of the biotic interaction. And we perform a couple of essays that I will show you some of them here that are already published. One, it's an arthropod bioassay using pillbugs or roly-polies. Basically, we starve roly-polies and then we put them, we made a choice experiments offering them wild type or the mutant plants. And after 24 hours, we quantify the damage performed by these arthropods. And this is what we observe. So this is before damage and this is after damage. And you can see here that the area removed by the arthropods, by the pillbugs, was a lot higher in the mutant compared to the wild type. And this is just a quantification. The area after 24 hours is almost the same in the wild type, however, is smaller in the mutants, okay? Showing that these guys prefer the mutant plants. And it's very likely that that's a reason of the depletion of these terpenoid compounds. In addition, we also performed some assays with some bacteria. We tried E. coli, gram-negative. We didn't see any phenotype there, but we did see a phenotype for Bacillus subtilis. So basically, this is an overlay assay. What is this? That's there you grow, a culture of Bacillus, a uniform culture on a plate. And before growing it overnight, you put a drop of, this is an ethanol extract of the plant, can be either a wild type plant or the mutant plant. And then you observe the level of inhibition induced by that extract, okay? And after overnight, what you see is that the wild type extract, induced a heavy repression of the bacteria growth here. However, that inhibition was smaller by the mutant extract, again, suggesting that the all body content actually has compounds that control somehow the biotic interaction. Okay, so we were really happy at this point because we answered a long-standing question about regarding the all body function. So we showed here that all body functions are involved in the biotic defense of liverworts, and in addition, that the all body function is controlled by this transcription factor. Yeah, so we finished our story proposing a model for our TF, dynamic of all body formation. So close to the notch, all the cells are expressing our transcription factor. Somehow, we still don't know how, the expression fades away in neighboring cells, in some of the cells, and in others keeps high. And those cells that are the ones that actually form an oil body. Okay. So all these, our main conclusions are that all bodies are part of the biotic defense response of liverworts, that this transcription factor is a molecular switch controlling all body formation in all bodies. Okay, so with that, I would like to show you a couple of some more slides, to tell you how are we moving forward. So this is a picture of a review that we are publishing now in Journal of Experimental Botany, where we basically made a big search of what is published and what is not, and mapping and tracking the phenotypes related to the type of oil body. I mentioned you that there is a big diversity of oil bodies. So for example, most of this group that belongs to a class of Marchantia related species called Marchantiopsida, all of them differentiate their bodies in one special cell, okay? And is one large oil body. Because of that, they are in blue. There are some exceptions with some species that do not form oil bodies, like this one here, Blasia, okay, or Monocarpus or Sphyrocarpus. What we found there is that actually for those species that lack oil body, they also, and at the same time are reported their biochemical profile, they also lack sesquiterpenoids, okay, what match what we found in our Marchantia mutant. And then you have species, all these, that belongs to another group called Hungermaniopsida, that has several oil bodies in all their cells, okay? They don't have any special cells. So you can see that the profile is different, no? So now what we are trying to see, to show actually, is if the expression and maybe some polymorphisms associated with this transcription factor is responsible in part for the chemical diversity observed in liverworts. That's of course a hypothesis that we haven't answered. What do we use for that? And we used, again, the 1kp and 10kp data, okay, that will tell us, okay, where are the expression, what is the expression of those genes, if those species are or not expressing these transcription factors, and even more importantly, if that gene being expressed there has polymorphisms, maybe within the conserved domain. Okay, so this is the way where we move into. Okay, we are trying to find, again, polymorphisms at the protein level, and for some cases where the genomes are published, we are trying to analyze the presence of gene duplications or gene deletions that can also be possible. Okay, so with that, okay, so we, as I mentioned you before, we are also working in parallel with Arabidopsis, and so what we are trying to do is actually using overexpressing the Marchantia gene in Arabidopsis to try to complement the phenotype observed for some of these single mutants, single or higher order mutant that we have in the lab. Well, so far I won't talk to you much about it, but you know, we want to see to what level the Marchantia gene is able to restore the functions that are absent in some of these Arabidopsis mutants. So with that, I'd like to thank you, this space, and all my lab. Much of this work was done by Facundo Romani, and now he's working in Cambridge doing his postdoc at Jim Hassel's lab. Juan Ignacio Tolopka and Melina joined my lab and are heavily working on this as well. And I mentioned a couple of international collaborators. John Bowman was a great support to us because give us the know-how to work with Marchantia and transform it, and Miltos Siantis, as I mentioned you before, help us to move forward this project. So with that, I'd like to thank you, and we'll be happy to take any question.
Thank you, Javi, for the very nice presentation, very interesting data you had. So I will open for questions. I will say a few words in Portuguese. Pessoal, se vocês quiserem fazer as perguntas em português, em inglês, em espanhol, eu acho que qualquer uma das possibilidades é válida. Muito provavelmente, mesmo em Javier é capaz de entender. Se não for, a gente pode traduzir também, mas não deixem de perguntar, porque só...
Vou falar em português, tá? Acho que fica melhor
Tudo bem,
perguntar.
maravilha, tranquilo.
Gostaria de agradecer, né, ao professor pela apresentação dele. E eu gostaria de perguntar, é que eu não entendi muito bem, porque tem um momento que ele acompanha o corpo de óleo, né, o corpo oleaginoso, numa célula só ali no momento. E aí tem alguns momentos que ele fala que uma célula só que faz isso a partir do desenvolvimento. Eu não entendi se é sempre uma célula que faz isso ou tem outras plantas que todas as células fazem isso, ou que regula que só essa célula vai fazer isso.
O que regula somente uma célula expressa?
Isso, não sei se eu entendi certo, né? Porque tem ali um momento que ele acompanha só uma célula, né? Aí as outras a gente vê que não tem.
¿Entendiste, Javier? La pregunta, Flavia, você me corrija si no es exactamente eso, mas lo que Flavia está preguntando es que en algún momento vos mencionaste que los cuerpos oleosos en diferentes especies pueden ser pocos o en algunas células especializadas o en todas las células, ¿no? La distribución, el patrón, digamos, de cuerpos varía en diferentes especies. Si se sabe qué es lo que determina eso, que haya un single cell omnipresente en todas las células. ¿Es eso, Flavia?
Yeah. Sim, obrigada.
Well, I think, Flavia, you did a great question, because actually we are behind that in the sense that we want to try to explain the diversity of all bodies and the diversity of patterns with maybe some genetic polymorphisms that we are trying to find, So we have some candidates, and we are now looking into the sequences, mainly obtained from transcriptomic data from 1kp and 10kp. So to see if polymorphism is at the protein level, because we don't have the genomes. Yeah. In the case that we have the genomes, we can go to the genomes. And in the case that we have the genomes, we can also look into the promoter region, because maybe the difference is at the expression level, not at the protein. So that can be a challenge, because we are limited there in that case by the availability of the genome. So you did a great question, and we are, now we are working with another species here in the lab. It's Blasia that lacks all bodies. So, and we are trying to complement that with some of the candidate genes. Okay, so to see, our idea is the presence of, let's say, one of the genes is able to restore the formation of all bodies. Yeah? So I think you are making a good question, because it has two sides, that question. One is regarding the type of all bodies, how many and where, and the other is if the type of foreign body also change the biochemical profile of that species. So we are, behind that.
Okay, thank you. Thank you very much.
You're welcome.
¿Tem alguma outra pergunta aí pendente?
A gente pode ir fazendo para motivar os alunos.
Então, I have a couple of questions. One, somehow you have already answered, because I immediately thought about what happened if you put this gene into Arabidopsis or any other angiosperm. And I would like to know which Arabidopsis mutants are you trying to complement?
Well, so we have single, most of them are single mutant. There is only a triple mutant described for Arabidopsis. Okay, that's a high order mutant that has three HDC knockouts. And we are trying to complement that as well.
Those are mutants for the enzyme terpenoid biosynthesis on
those are mutants for the
HDC genes. Okay.
Genes,
okay.
Yeah.
So they are deficient in jasmonate response, for example, or in abiotic.
Well, so most of them, so for example, the HDC1 of Arabidopsis has a hypocotyl phenotype. Okay? The triple mutant has a branching phenotype. Okay, so we are basically transforming and see what do we it's hard to predict if
okay.
but, you don't know a priori what are you expecting, if it will be able to complement it or not. Okay,
so you are trying to complement morphogenetic phenotypes so far.
So we also were interested in terpenoids, but you know that Arabidopsis is really not very good emitter of terpene compounds. So it's very hard to measure terpenoids. What you do, but in our lab, we don't have the expertise to do it. So yeah, the question is, we are not able to measure terpenoids in Arabidopsis yet.
And the other question is regarding the specific function of this gene. Okay, it's clear that it's a transcription factor, but which are the genes that this transcription factor induce or repress? Are they related mostly to the vesicle formation, the structure, or inducing the enzymes for terpenoid biosynthesis?
Yeah.
Because, I mean, you have seen a correlation and a spatial correlation, but this is not clear. What exactly is this transcription factor doing? Which are the targets, the promoter targets?
So one of the problems with the HDC that was shown for Arabidopsis is that you can find HDC binding site in 50% of everywhere. So it's really hard to predict what are real binding sites and which not, you know. So that is a little tough because you have to try them experimentally, basically, at least to say which were the relevant one. And well, you know, what else can I tell you? I mean, I think, well, yeah, I can hear you. You're muted.
Have you identified the biosynthetic genes and tried to follow the expression of those ones?
Yeah. So as I mentioned in the presentation, there are a couple of terpene syntheses that were downregulated, and we haven't really done much about it, you know. There are, you know, terpene synthesis, you know, in general, in the genome of Marchantia, what is described is that the specialized metabolisms, enzymes, and genes have suffered duplication events. They are usually larger than the families in Arabidopsis. No? And then you have this issue that in bryophytes, you have these terpene syntheses that are clearly coming from lateral evolution events, coming from fungal or even bacteria. Okay, those are called microbial TPSL. So far, we haven't done much. There are a couple of groups with a lot of expertise in terpene synthesis working on them, but we haven't really done much about it.
Right.
Javi, can I say something, Magda?
No, go ahead.
Okay, there's a question in the chat from Felipe, one of our students. I think it's a very interesting question. He said that he was wondering why these plants accumulates or produce so much oil bodies, and whether they are also used as an alternative energy source.
Well, so that was one of the hypotheses. You know, liverworts are being studied since 200 years ago. There is a lot of course, biochemical research done in the early 1900s. And that was one of the ideas. So far, we didn't find any limitations in terms of energy or growth, obvious growth phenotype in the mutant. So it's hard to answer your hypothesis. We don't have data for that. It is possible. I mean, and actually we tried several abiotic conditions or treatments, but still, you know, there are some guys that are saying, well, you know, maybe it depends on the conditions, and it's true. I mean, so that's a good idea. We haven't tested that, but it's true that they are all compounds that can be useful also for as a energy allocation place or compound.
Okay, great. Thank you. I have one question that I would like, while we wait for other students to ask. For me, was really striking when you showed the comparison between the number of genes in each gene family of transcription factors between Arabidopsis and Marchantia. And it's not that in Marchantia, the number is reduced. I think most of the time there are single genes in each family. So I was wondering regarding the functions of those genes. Do you know whether in Marchantia, because of this reduced number of transcription factors, whether they are more related to have like multifunction, like one particular transcription factor might be able to perform more functions or to control more biological functions. I will give an example. I noticed that the NAC transcription factors, it's only one NAC, and in Arabidopsis you have NACs involved in responses to abiotic stresses, but also involved in cell wall formations, like completely different biological processes, but those are different groups of NACs. And in Marchantia, you have like only a single NAC. So is it possible that this NAC is performing a lot of functions, or how do you see this?
Well, so there are already some papers published about it, no? So first, I have to tell you that we published a review, you know, taking all the information that we have gathered during this time. What we observe in general is that the transcription factors that control developmental responses clearly they might have, you know, like a heavy selection pressure on them, and they usually do the same here and in Arabidopsis. Okay. On the other side, when you have like this one, like my transcription factor, the C186 transcription factor related to the environmental response, they might do different things in different plant species. Okay, so however, so for example, there is a case of the phytochrome. Phytochrome is, you know, it's not a transcription factor, but it's a master receptor of light quality. The Marchantia phytochrome does a little bit of both phytochromes, A and B, of their Arabidopsis ones. Okay, so it's able to complement some functions of the phytochrome E mutant and also some other functions of the phytochrome B mutant in Arabidopsis. So what else I have to tell you? Then you still have another option, no? That is you can complement Arabidopsis mutant, and they may not do the same, okay, or they might, and that's that's something that you have to do. It's an experiment that you have to do in order to show it. You don't have another chance. Basically, it will depend on how that transcription factor interacts with the gene regulatory network, okay? And so far, I'd say that it's hard to predict.
That was another question of mine. Do you know about like protein partners? Because I mean, sometimes the transcription factor, it might have a different function depending on which protein partner that it might have. Is there any like study on transcription factor for Marchantia about the protein partner?
No, not in Marchantia, or not at least published yet. So that would be interesting, you know, to do at least a yeast to hybrid screening or something like that, or even a proteomic approach, but we haven't done. I believe that there is a project, you know, a multi-lab project trying to do something like that, but so far it's not always obvious. That's what I have to say. You know? What the prediction is not always something that you can do in a linear way. Let's say if in Arabidopsis do this, in Marchantia will do this. No, it won't work.
Okay. And my last question, then I'm sorry guys for the taking the full control. You mentioned that, I mean, the low redundancy of the Marchantia genome, of course, that might be an advantage when you're skipping the problem of having the redundant functions between genes. But I was wondering on the other side, I mean, as a drawback, do you see like, because it's a single gene, a lot of gene families, do you see problems like when you're knocking out a gene, then you have like a deleterious effects?
Yeah. So there are not much papers, you know, but there was a case for a CYP12A mutant that they didn't recover any mutant. So that is lethal. No?
Yeah.
So you can have that problem. So that can be a drawback. Okay.
Yeah.
Tell you something else. My guess also is that we could potentially use the liverwort phenotypes to uncover new roles in angiosperms. Okay, because sometimes, you know, in the angiosperms, the phenotype is basically masked by other guys that are doing the same. So hopefully, so for example, here my prediction would be that maybe HDC genes in angiosperms are controlling the terpenoid phenotype. And we haven't been able to do that, maybe because nobody looked, or maybe because of the redundancy. You know? So yeah, we are interested in that as well.
Okay, thank you very much. Does anybody else have any questions to Javier? We still have time. No? Okay, so if we have no further questions, I would like to thank Javier once again. It was super interesting, your talk, your data. I will be following your work as I was doing before. And I thank you very much in regards of all people involved from Magda, Leandro, and I would like to thank you. And yeah, and thanks all the students for attending as well.
Wonderful talk, Javier. I didn't know about your work and I really love it. Yeah. Congratulations.
Well, thank you so much, guys. And we are open here for future talks. And I will steal this idea about the seminar series and I will invite you. Okay?
Yeah, great.
Great.
Okay.
Thank you very much.
Thank you. Thank you, Javier.
Thank you, Javier.
Bye-bye.
Bye,
Bye-bye.
guys.
Bye. Tchau, gente. Boa semana para todos.